Well, hello and welcome to lecture 13 and 14 regarding cytoskeleton. Now, before I start cytoskeleton, there's something which I need to say, and that is these two lectures, cytoskeleton 1 and cytoskeleton 2, are quite unique in a sense that there are no minimal questions from them so far. They maybe give some questions in the upcoming years, but so far there are no minimal questions from them. These lectures are quite massive. Each of these lectures are about 67 or 68 pages, but in truth, in the exams, there are so far there haven't been more than 2 or 3 direct questions from cytoskeleton, and those questions, even those very easy questions usually, are coming repeatedly from a specific slides from these lectures, especially the second cytoskeleton lecture, which I have hardly seen questions coming from it. Maybe only 1 or 2 slides in the second lecture I think is important, and there is a potential of giving question from them. Although, as I said in the very first video, they may change everything. Like there is a possibility that next year they change the whole system and they only give essay questions, like 5 essay questions from cytoskeleton. But I'm gonna talk about cytoskeleton lectures based on the hypothesis that they are going to move with the cytoskeleton as they've done so far. And if they do so, which is a high possibility, it's going to be a very easy lecture with only a few slides to understand and remember. So let's get to it. First of all, the components of cytoskeleton. There are 3 main components. These 3 components, these 3 filaments, make the skeleton of cell, the cytoskeleton. And these pictures here, these pictures that you see here, they are important. One of those few questions that I mentioned is that they give like the picture for microfilament, and they ask, what is it? Is it the microfilament, microtubule, or intermediate filament? So these pictures are important. These 3 pictures here are important. And as we said, there are 3 types of cytoskeleton filaments: microfilaments, microtubules, and intermediate filaments. The subunits of microfilaments are actin filaments. So microfilaments, their subunits, their monomers are actin filaments. And as you can see, these filaments are on the edges, are spread on the edges of the cell, mainly because they are very important for the cell-cell or cell EC extracellular matrix junctions. So they are important for cell-cell contact and cell ECM contact. That's why they are mainly, that's the main reason they are distributed like this in the edges of the cell. And in case of microtubules, as you can see, they form a spindle kind of formation. And that is because our centrosomes, which are the organizing center of centrioles within mammalian cells, our centrosomes they have centrioles, and in the centrioles they are microtubules. And as you probably know, these microtubules, these centrioles, are responsible, one of their responsibilities actually, but one of the most important ones, is the division, is during division of the cell of information of the mitotic spindle. And that is why they look like a spindle. Mainly, that's why they look like a spindle in the cell. And their monomers are alpha and beta tubulins. So alpha and beta tubulins are the monomers of microtubules. Whereas in case of intermediate filaments, you can see these intermediate filaments don't really follow a specific structure. And that is mainly because these intermediate filaments, they have the greatest tensile strength among those other two filaments, and so these are the real skeleton of the cell. These are the ones which make cell very resistant to pressure, to mechanical stress. And you can see here it's written that their monomers, their subunits are various. But what you need to know about the subunits of intermediate filaments is that they are formed by different types of keratin proteins. So keratin is the word you need to remember, keratin. And here you can see their diameters. You do not need to memorize exactly their diameters, as long as I know, but what you need to know is that which one of them is the thickest and which one of them is the thinnest, so and which one is in between. So microtubules are the thickest filaments. Microfilaments are the thinnest, and intermediate filaments are in between. You can think of intermediate as in between in case of diameter. Now, the lecture starts talking about each of them in more detail. But what you really need to know are the very basics of the very concepts of these filaments. Like the structure of microtubules, they form hollow tubes. So when the monomers, when the alpha and beta tubulin monomers form dimers and they form the microtubule filament, they form it in a fashion that there will be a tube with a hollow center. So the center of the tube is empty, there's nothing in it. And that if it consists of how many columns, that is, I don't really think that's important, that 13 columns form this tube. And the diameter, I said the important thing about the diameter of microtubules is that they are the thickest. And subunits, as we said, tubulins. Now, the main functions are the ones that are actually important. Maintenance of cell shape is the function of all of these cytoskeleton filaments. That is why they're called cytoskeleton. They maintain the shape of the cell. So that is one thing they have in common. Now, in case of microtubules, as we said, they're very important in chromosome movement in cell division, and you probably know it. Cell motility and organelle movements are also functions of tubulin, but they are not as much important as the chromosome movement in cell division and maintenance of cell shape. One other thing about the tubulins is that what makes these tubulin subunits different? What is the difference between alpha and beta tubulin? That is due to presence of GTP or GDP. So alpha tubulins, they have GTP bound in them, whereas beta tubulins, they have GDPs bound in them. And that is important. This the GTP or GDP binding is important both in treadmilling, which we're going to talk about, treadmilling of these microtubules, and both in the function of dynein, which we're going to talk about again. This is the motor protein. Now, details about intermediate filaments. They are fibrous. That is one structural prominence of these filaments that you need to know. They are fibrous proteins supercoiled around each other. So they're not two or like three. There are a lot of these fibrous proteins coiled or supercoiled into thicker cables. The diameter, as I said, you just need to know they are in between of microtubules and actin filaments or microfilaments, and that their subunits, if you remember, it was written various. Now it says one of the several different proteins of the keratin family. So the keyword here is keratin. You have to memorize keratin as the subunit for these intermediate filaments. And the functions, well, maintenance of cell shape, as we said, is common in all of them. And if you remember from the lecture about nuclear pores, they form the nuclear lamina. So nuclear lamina is intermediate filaments. Lamina A, B, and C, they are intermediate filaments. And so forming nuclear lamina and maintenance of the cell are the two most important functions, and anchorage of nucleus and certain other organelles is also another function, not as important as the other two. Now, the last of them is the actin or microfilament, which is formed by actin subunits. Now, what is the structure of this microfilament? The structure is like DNA. It is a double helical structure. So there are two actin filaments which intertwine each other, and like they form a DNA-like structure. But do not mistake them, because DNA is formed by bases, by nuclear bases, but these microfilaments are formed by proteins. That's a very different thing, but they resemble the same structure. And their diameter, you need to know, again, they are the thinnest ones. Their subunits, actin, and their main function, well, maintenance of cell shape again, and the other very important function is in muscle contraction. We are gonna talk, especially when you're studying for histology, you're gonna study this muscle contraction in detail and the role that actin filaments play. But we're gonna talk about the basic things you need to know of actin role in muscle contraction. And then we've got cell motility, cell division, and cytoplasm streaming. Again, these are not as important as muscle contraction and maintenance of the cell shape. And so now that you know about each of these filaments, there's a slide which summarizes the main important features of each of these filaments. And this slide, from my point of view, is one of the most important slide of this lecture. So from this slide, one of the things that you're gonna understand is that microfilaments and microtubules are very alike. They have like differences, but their actions are very alike. Whereas intermediate filaments, they have very big differences. So let's talk about each of them. In order for microfilaments or microtubules to perform their action, they need the microfilaments need to bind with ATP, and microtubules, they need to bind to GTP. And what are their actions? Because you may think they are just like they're just like the skeleton. They're like the column of a building. They just stand there and they hold the place together. But that's not true. These filaments are constantly shrinking and growing in the cell, and other proteins, other motor proteins, are walking on them and transporting material. So when we're talking about the cytoplasm of the cell, it is not like any protein or anything just floats in the cytoplasm from one place to another. If these proteins want to move in the cytoplasm, they need to use roads, they need to use special transporters, special cars, for example. And these the filaments, these microfilaments, are the roads, and the motor proteins are those cars. And in order for those cars, those motor proteins, to drive in these roads, these microfilament, these filaments, the action, the binding of ATP is necessary for microfilaments, and binding of GTP is necessary for microtubules. But intermediate filaments do not bind to nucleotides, and that is mainly due to the fact that there are no motor proteins for intermediate filaments. So to think that there are cytoskeleton filaments that basically act as the column of the building. Intermediate filaments are those columns. Intermediate filaments are like exactly the skeleton of a human being. They hold things together. Whereas microfilaments and microtubules can also act like roads within the cell. And you can see intermediate filaments, they have a very great tensile strength. So the resistance to mechanical pressure, to all sort of pressure, they work as the real skeleton. And this part isn't that important, but this part about the dynamic of each of them, you can see microfilaments and microtubules are highly dynamic, whereas intermediate filaments are less dynamic. Again, common sense. Intermediate filaments, they're just there, they're just being the skeleton on which everything else is being built, whereas microtubules and microfilaments, I said they act like roads. Now in our cells, roads themselves move. So in contrast to the example I said, where it's the car moving in the road, in case of microtubules and microfilaments, in case of the roads in our cells, the car and the road together, they move. Both of them are dynamic. So microtubules and microfilaments are highly dynamic, and they're also polarized, and being polarized is a necessity for being dynamic, and we're going to talk about why being polarized is a necessity for being dynamic. And again, common sense, you can see intermediate filaments are unpolarized. The name of motor proteins, the name of those cars which I just said, for the actin filaments, for microfilaments, are myosin. So myosins are motor proteins for microfilaments, whereas kinesins and dyneins are motor proteins for microtubules. So we said microtubules and microfilaments themselves can grow or shrink. Basically, they can move with this growing and shrinking in the cytoplasm of the cell themselves. So to put it in better words, it is called dynamic instability. And dynamic instability is the periodic growing or shrinking of the microtubule or microfilament length due to polymerization or depolymerization. So if, for example, we take the growing part, we have polymerization of that side which is growing. So polymerization will cause the growing of that side. And if we've got shrinking of one side, it is due to depolymerization. These monomers are leaving. So polymer to monomer, depolymerization. And that is how these filaments themselves move. And just keep in mind that, as we said, both microtubules and microfilaments are polarized. Don't mistake it with polymerized. They are also polymerized, but they are polarized. So there is a positive end and a negative end, like a battery. There is a positive end and a negative end to these filaments, and this polarity will help them to shrink from a specific site and grow from the other side, and that will lead to treadmilling of these filaments. And treadmilling is both is a character for both microtubules and actin filaments, and that is when the length of the cytoskeletal element which is the microfilament or microtubule, the length of the cytoskeletal element doesn't change, but the position of each molecule is changing in time. So that whole dynamic instability which we just talked about, from one end, depolarization from one end and polarization from the other end, will lead to treadmilling, where the length of this road, the length of this filament is constant. But since the position of monomers are changing, it is moving. So as you can see here, monomers, this is a microtubule, and so these monomers are alpha and beta tubulins. Monomers are getting added from the positive end. Yes, they are also leaving, but if you pay attention here, the ratio of them getting added is more. So the net adding of these the net polymerization of the positive end is bigger. So the microtubule is growing from this end, and in the same time it is depolarizing, it's shrinking from the other end. And that's very genius, because this road of ours in our example is moving, and imagine something is on it, a protein, a motor protein like a dynane is on this microtubule, and dynane wants to go from position A to position B. So if dynane was to walk all the way by itself, it would take a very long microtubule to do so. But when the microtubule itself is also moving, it just helps the movement of this motor protein way more efficiently. So one more thing about this whole treadmilling thing is that the polymerization occurs in the site with a lower critical concentration, and depolarization occurs in the site with a higher critical concentration. What is the critical concentration itself? I really do not think that you need to know it. And now that we know about treadmilling itself and the movement of these microtubules, let's talk about the movement of these motor proteins on microtubules. And in case of microtubules, we are talking about dynanes and kinesins. They are very alike, dynanes and kinesins. Their main difference is a bit difference in the structure, as you can see, but the main difference is that they move in opposite sides. They move in opposite directions on a microtubule. And the way that they move is that they have two heavy chains on the lower end, on the end which attaches to this microtubule, and with these two heavy chains basically act like feet. So these proteins walk on these microtubules, and as the professor himself said, pay attention that these proteins are not running on the microtubules, they are walking on the microtubules, which means at any given moment you cannot have both of these legs detached from the microtubule. So the way they move is that one leg is attached to the microtubules, and that is when the other leg is detached, and when the other leg is attached, the opposite leg will be detached. So they are walking. You cannot have a moment in which both of these legs are detached from the microtubule. And this moving is due to phosphorylation and dephosphorylation of these heavy chains or legs of kinesin or dynanes. So due to phosphorylation and dephosphorylation. And this process itself is a bit complex because it is the phosphorylation of one leg which causes detachment of the other leg, and the opposite way around. So I again do not think that they're gonna ask about how exactly is the process of phosphorylation But again, it's a possibility that they change everything. They ask you all essay questions about these parts. But I'm gonna go with the assumption that they are going to repeat like the previous years. One more thing about this lecture in a specific is that you basically need to know that cilia and flagella are microtubules, are formed by microtubules. Now, where we can find the flagella in, for example, sperm, and when the tail of the sperm, and cilia, we can find it in, I don't exactly remember, but I think esophagus. I'm not sure, but cilia are very actually flagella are very much longer than cilia, and that's the main difference. But the thing that you need to know is that cilia and flagella are made by microtubules. And so we go to the second lecture slide. Again, this lecture is by Professor Mathias, and honestly I only found one slide in this lecture which they gave questions. They usually don't give questions at all, and if they do, this is the one slide that they give question about, and the concept of this slide, not even the details and the steps of this muscle contraction actually here. So I'm just gonna talk about this one slide. Again, if you have time and you wanna make sure that you'll do perfect in the exam, you can read all the lecture by yourself. I'm not promising anything that there won't be other questions from other sides of the lecture. This is just my suggestion. So back to the lecture slide. This is about the myosin action on actin filaments. So we said the motor proteins for microtubules are dyneins and kinesins, and the main difference is that for them is that they move in opposite directions. And the motor protein for microfilaments, or the actin filaments, which microfilaments are made of, the motor protein for them are myosins. Now, in our muscle cells, we're gonna have these the thick, thin filaments which are responsible for the contraction of our muscle cells. And the way our muscle cells contract is that these myosin proteins, these myosin motor proteins or filaments, they have a head, and their head will attach to the actin filaments and will simply grab them and pull them, and then this will happen again, and that is the basis of muscle contraction. And how does this attachment, pulling and detachment and the cycle works is due to, well, yes, you guessed it, phosphorylation. So again, I don't think they ask you about the details of phosphorylation, but since we're only talking about this slide, let's talk about these details. It's not that hard to understand, and that is basically we have our myosin in a bound form to actin filaments, and when ATP binds to the myosin, conformational change occurs in the myosin. Myosin will detach from the actin filament, and when ATP is hydrolyzed to ADP and phosphate, inorganic phosphate, the myosin will reattach to actin. Now that is the detaching and attaching parts. How is the pulling happening here? How is the myosin pulling these actin filaments? When this inorganic phosphate leaves, when the inorganic phosphate leaves the myosin, myosin will undergo furthermore conformational change, and that leads to pulling the actin filaments, and at the end contraction of the muscle cells, and this cycle will happen over and over again since the calcium signal is over, which we talked about the calcium signal and what happens in the calcium homeostasis. That was basically everything that I think you needed to know about these two lectures. One other way that they have to ask questions is that they mix questions between these lectures and the lectures about cell cycle or cell-cell contact. So you have to also have knowledge about those lectures if they ask questions. Usually that's the way they ask mixed questions. Like, for example, what type of filaments do we have in desmosomes and hemidesmosomes, and what type of filaments do we have in case of the adhesion belts? So one of them is intermediate, one of them is microfilament. Or when we talk about this cell cycle, treadmilling and the dynamic instability are also gonna be in the cell cycle. So that is another way for them to ask questions. But basically that was it for these two lectures, and see you in another one.